Hello listeners and welcome to the Dr. GPCR podcast. Did you hear about the Dr. GPCR University? It's the ultimate learning platform within the Dr. GPCR ecosystem. Here you can expand your knowledge, connect with experts, and stay informed about the ever-evolving world of GPCR research. We offer both live and on-demand courses covering everything from GPCR pharmacology data analysis to drug discovery strategies. Our courses, like those that focus on using Prism, help you master the pharmacology data analysis framework, navigate the complexities of GPCR research, and improve your work's quality, reliability, and efficiency. Join us and elevate your GPCR expertise today. And now let's dive into this episode.
Hello, everyone. This is Yamina from Dr. GPCR, and welcome to yet another Dr. GPCR podcast episode. And this very early morning, I have with me Dr. Ian Chronis, a very recently minted Dr. Ian Chronis. Ian, welcome to the podcast.
Thank you, Mina. Very nice to meet you, be here on the Dr. GPCR podcast for the first time, and excited to get a chance to chat with you.
Very excited to have you on. Before we hit record, you had mentioned that you very recently were awarded your Ph.D. degree. So let's start at the beginning. If you can introduce yourself, tell us who you are and what is it that you do.
My name is Ian. I am a postdoc at the University of Michigan. Very recently, actually, as of right after this, I'm going to go have my I-9 meeting to officially be declared a postdoc at the University of Michigan. I just finished my Ph.D. there in the lab of Dr. Manoj Puthenveedu. There I study adrenergic and proton-sensing receptors. And yeah, I finished my Ph.D. very recently. I just defended in June and finished up my last dissertation revisions in September. So very recent doctorate.
Fantastic. No, this is fantastic. I've been trying to get Manoj on the podcast for a very long time. And every time I bump into him at a meeting, he says, yes, I got it. But I'm assuming he's very busy.
Yeah.
So
Yeah, he has a lot going on.
Perfect. So did you always know that you wanted to be in science? Was there any inclination as a young child or teenager?
That was definitely the primary plan from when I was pretty young. I grew up in a family where a lot of my family were in the medical field. And so I'd been exposed to that when I was fairly young. And I thought that if I was going into science, I was probably going to go to medical school. But there was always a reasonably strong chance I would diverge from that. When I first started undergrad, I actually started taking introductory courses for a classical history degree. So I was taking courses in ancient Greek, but I did eventually decide that wasn't quite right for me. And due to some course conflicts, I ended up moving away from the introductory sequence there and dedicating myself more fully to, at the time, it was a chemistry degree. And by the time that I finished my degree, I still wasn't sure whether I was going to medical school or whether I was going to do a more research-focused degree, and eventually decided that research was what I wanted to focus on. I had a really good research experience while I was an undergrad. This was at the University of Chicago, working on protein engineering and using directed evolution. And I thought that was really exciting. And after that, I did a master's degree at the University of Oxford in pharmacology, trying to move myself more toward a biology direction. And once I had finished that, I had kind of decided that, yes, I wanted to be in research. Medical school was not the route that I wanted to go for myself. And so then I was applying to PhDs in pharmacology primarily.
That's awesome. Considering that you very recently finished your PhD, when you were looking for a PhD position, what were the steps that you took? What were your criteria? And then I'm going to ask the same thing for the postdoc as well.
Because I have always been very interested in a lot of different things, and I found it a little bit difficult to narrow myself down to one particular focus. So I have had a habit of, at every step along the way, looking for the thing that will still afford me the widest variety of opportunities. and the widest variety of different experiences to have, things to learn. And so when I was looking for PhDs, the thing that was really valuable to me was places that had lots of different things available, lots of different faculty in lots of different fields, expertise across a broad variety of subjects. So one of the things that really attracted me to Michigan specifically was when I visited, the number of professors available that I could have worked with or had internal collaborations with is enormous. And the variety of expertise is, and even if like Michigan doesn't have the one person who is a world expert on something, there's somebody who has a connection to them. So that was a thing that was really valuable to me, because at that point I still didn't know exactly what I wanted to do. And so part of what I wanted to do is go to the place where I knew that whatever I ended up deciding to do, I would have somebody there who was going to be able to teach me, and I was going to be able to interact with who was at the top of the field.
I love it. I love it. Which leads me to my next question. How did you get into studying GPCRs? And it was did you find GPCRs or did GPCRs find you?
It was mostly an accident. When I was interviewing in the pharmacology department at Michigan, one of my interviews was with Manoj, who is an expert on GPCR trafficking. And during our meeting, my interview, he was telling me about some of the work that was going on in his lab, and I had looked up a little bit of it previously, but I had not at that point realized that GPCRs could ever signal from compartments other than the plasma membrane. I was familiar with them at a basic level, but that specific idea of an entirely new avenue of receptor signaling that responds very differently to drug treatment, and this spatial localization and spatial determination of signaling complexes was something that was really interesting to me, because it seemed like something that was a new dimension completely outside of what I had learned in a class. And so when I eventually came to Michigan, I asked Manoj if I could do a rotation in his lab. That was my first rotation in the pharmacology department. And between finding the GPCRs as being such interesting targets from a cell biological perspective, but then also knowing that if I'm working in a GPCR field, there is always an avenue toward that more translational aspect. There's such ubiquitous drug targets that even if I'm thinking, okay, I'm working in a cell biology lab, I'm working in a cell biology project, which I think is very exciting. You always know, there's always that hint of, but this could really matter. This could really matter for developing new therapies, because they're such good drug targets. And so, I did my first rotation in Manoj's lab. I started learning about GPCRs, and then that lab had a lot of connections to the lab of Dr. Alan Smirka, who's also at the University of Michigan. So then I did a rotation in the Smirka lab, and by that time, pretty much all of my graduate level training was in GPCRs, and so I ended up going back to Manoj's lab for my dissertation research, and I probably am going to study GPCRs in one sense or another for the rest of my career. If I can get that to work, I'm going to.
I love it. I really like it. One thing that I kind of envy from the US system is this ability to do rotations. So I studied at the University of Montreal, and you didn't have the opportunity to do a rotation. You finished your bachelor's, and if you wanted to get into a master's or a PhD degree, you needed to pick a lab, and that was it. And switching a lab after a year or two is very difficult. And how do you explain that as to, oh, things didn't work out, or I didn't like the project, or all of these things? So having the ability to spend some time in the lab and getting a sense without committing fully yourself to one project or one lab specifically, I think it's a really nice thing. You mentioned GPCR signaling beyond the plasma membrane, and I think it's an interesting topic, because there's a lot of work that's coming out on that, but I'm not sure if if we can, as of now, exploit it as from a drug discovery perspective. So let's track back. I want to hear a little bit more about your PhD project and how it connects to this particular topic. I have a couple of other questions, but let's start with your PhD project. I'm really curious as to what did you work on?
I had two major things that I was looking at. One project, which was the one that I began when I first joined the lab, focusing on the beta-2 adrenergic receptor. And then later on in my PhD, actually starting about a year ago, I also took on an additional project working on the proton-sensing receptor GPR65. So the original project, what I was originally trying to look at with the beta-2 adrenergic receptor, our lab primarily worked from Drs. Rachel Vistine and Shana Bauman in the lab had previously identified phosphorylation by pKa of a specific pair of sites in the receptor C-terminal tail as regulating certain aspects of its signaling and trafficking. So the beta-2 adrenergic receptor, like many receptors, is internalized after acute agonist activation, and it can signal from internal compartments. That's been known for a little over a decade now, that the receptor can be active and induce signaling cascades once it's at endosomes. And they had identified that phosphorylation of the receptor by pKa is required for this signaling behavior. And our hypothesis was that there is some scaffolding protein that is associating with the phosphorylated receptor at the endosome that is required for promoting signaling. So I was originally doing proteomic work using a proximity labeling strategy to try and identify new interactors of the receptor, specifically ones that would regulate its ability to signal from the endosome, ideally in this phosphorylation-dependent manner. I will say that's not what I ended up finding. I did not end up being able to identify this mystery protein. I spent a long time trying to determine what that might be. But in the process of doing this proteomic work, I did identify an interaction of the beta-2 adrenergic receptor with 14-3-3 proteins, a class of scaffolding proteins. They're primarily phosphorylation-dependent scaffolds. So this was something that I was kind of interested in, and I thought might be what I was looking for. It didn't end up having any effect on the intracellular signaling behavior that I was able to discern. And actually, that had been an interaction that had been identified before by one group. But so I then investigated what 14-3-3 proteins might be in fact doing to regulate receptor activity. And what I seem to have identified is that they associate with the receptor primarily at the plasma membrane and sterically block it from interacting with other effectors, G proteins and arrestins, presumably by binding to the intracellular face of the receptor and sterically inhibiting its association with different proteins. So that was one of my projects looking at beta-2. But then the project that I've been working on the proton-sensing receptor is actually, I think, is a little bit of a more exciting, innovative project. That's also based on some previous work in the lab done by Dr. Loida Morales Rodriguez, who graduated from the lab last year. And what she had identified is that this proton-sensing receptor, GPR65, constitutively internalizes, and that it does so irrespective of its ability to be activated. So even if you make mutations that block the ability of the receptor to signal, it will still internalize the endosomes. And that's, of course, really interesting for a proton sensing receptor because the endosome itself is an acidic compartment. So unlike a lot of receptors, when we talk about signaling from intracellular compartments, there's always the very real question of, okay, but how does the agonist get there? And that's fair. For some, they're hydrophobic and they can just flow through. Other times they get transported. But it's always a question. You always have that question about ligands actually being able to access the receptor. But for a proton sensing receptor, that's really not a question because the endosome that the receptor is moved to is itself sufficiently acidic to activate signaling from the receptor. So we thought that this was both was a really interesting receptor to look at intracellular signaling because we know that that can happen in a physiological system. And you don't so there's no question about whether the receptor is capable of being activated at endosomes in real cells. And so having seen that it constitutively internalizes, what I've been trying to identify since then is what are the mechanisms of this constitutive internalization, and is it using canonical mechanisms of GPCR internalization machinery in an unusual way, or is it using novel internalization mechanisms? And so far what I've shown is that it does not require GRKs and does not require arrestins. So it does appear to be a novel mechanism of internalization, but still trying to figure out exactly what it is. I've shown what it's not, but still working on identifying what scaffolding proteins might be involved. And I have also shown that it is dependent on phosphorylation, but don't know what kinases are involved. So that's my current work.
I love it. So while you were mentioning GPR65, I remember that there's a company actually in the UK, in Oxford, called Pathos Therapeutics.
Yes.
And they're focused solely on GPR65 as an immunology target.
Yeah, it is a very important emerging target. It's part of the NIH's Illuminating the Druggable Genome project. It's one of the targets for that, but it's been designated in their class what they call T-bio, indicating that we know a decent amount about the biology, not nearly as much as we'd like, but there are no really good ligands for it, either activators or inactivators yet. But as you said, this company, a couple of other ones, are beginning to come out with chemical modulators, and then maybe not for GPR65, but for the other proton-sensing receptor, there are some other groups that have started developing positive and negative allosteric modulators of the receptors. So we're only just starting to get small molecules that can really act as, well, in my hands, I would think of them as being tool compounds, but then ideally, because it is an emerging drug target, hopefully some of those will eventually be useful therapeutics.
Absolutely. So what some of the questions, I find these proton sensing receptors really interesting because you need to be able to measure their activity.
Mm-hmm.
And obviously, since it's a proton sensing, you need to lower the pH. And so how do you reconcile, what are the, from a practical standpoint, what type of assays can you run and how much you would need to modify, for example, your assay protocol compared to beta adrenergic receptors, which let's be frank about it, beautiful receptor, a lot of tool compounds, and for some reason it always works, or most of the time, and that's everyone's positive control. But what were some of the challenges that you had working with GPR65?
Well, fortunately, Dr. Morales-Rodriguez had kind of optimized some of these assays a little bit by the time that I got started working on it. It's a little bit more cumbersome, but most of the time you can just make up media, pH the media to different levels. The receptor is pretty much totally inactivated at what we think of as a physiological media of 7.4. So a lot of the pre-made cell culture media that you can use doesn't have a lot of receptor activation, and you can make it slightly more basic to tamp down on what little amount of activation there is, and then just make up media to more and more acidic pHs. It's pretty much totally activated by a pH of about 6.4. And so after a little bit of trial and error, I've gotten to know like how to prepare media to do dose response curves with signaling assays and such. And we know that, and also, to some extent, what I'm particularly interested in is the constitutive internalization of the receptor. So I'm not that worried about having to acutely activate it. I just want to to some extent, keep it in a state where it's not signaling, at least from the plasma membrane. It's and what I'm looking at is the constitutive internalization. So, I don't even have to worry that much about activating the receptor. But then again, all of this is complicated by the fact that it will always have some amount of signaling and activation because it's going to these acidic compartments on its own. So when we talk about, we've activated the receptor or inactivated the receptor, for this particular example, well, we're talking about activation of the plasma membrane, the global membrane activation, and not actually the total amount of signaling competent receptor.
What do you think is, so I find it fascinating that this receptor internalizes constitutively. So what do you think it's at the plasma membrane where the pH, let's assume the pH is 7.4, so it doesn't have much activity. Whenever in a system where the pH goes down, then it gets activated, and then that's when the constitutive internalization happens, or the pH doesn't influence the constitutive internalization. I may have missed this.
I have a little bit of preliminary evidence that activation of the plasma membrane does increase the amount of internalization, but it really does seem to be internalizing irrespective of the extracellular pH. So the hypothesis is then that this is to some extent a like tonic signaling level, because it'll internalize, it'll be activated at the endosome, and it's also recycled back out. So it's exerting some amount of tonic tone, but then there's also a capacity for signaling at the plasma membrane if the extracellular pH does change, which it can. That's especially true in the case of the tumor microenvironment, which is why GPR65 is an emerging target in certain types of cancer. It's known to be upregulated in certain types of cancer, and generally have a pro-growth downstream effect. So we know that in the tumor microenvironment, the high glycolytic rate of the tumor, you get acidification of the extracellular media, and then you have this positive feedback loop where the GPR65, potentially other proton-sensing receptors, promote more growth, you get more glycolysis, you get more acidification, and so you have this positive feedback loop of acidification, growth, and so that's the signaling coming from the plasma membrane that is different potentially from the signaling that comes from endosomal compartments. And then you also have the other system where, as you mentioned, that GPR65 is of a lot of interest is in the immune system, which of course is intimately connected with cancer as well. But GPR65 is found on a lot of immune cells, and in those cases, in general, the receptor seems to depress immune function. So there are a couple of papers showing that in different types of immune cells, it reduces, say, the release of cytotoxic agents by immune cells if it's being activated at the plasma membrane by a change in extracellular pH. So when you put these two together, it has this pro-growth phenotype in a lot of cancers and this immunosuppressive action. You could imagine that tumor infiltrating immune cells are being suppressed while the signaling in the cancer cells is pro-growth. And I suppose it's not surprising that there are companies that are looking at it, trying to modulate its signaling in order to develop better cancer immunotherapy.
I love it. How cool is it? So if I just to paint a picture in my head, it's at the plasma membrane. Let's say at a normal pH, it still constitutively internalizes. When it internalizes, ends up in endosomes, where the pH is lowered, so presumably gets activated, does what it needs to do. So you still have this dual, quote unquote, dual function, yet the cells knew that you keep it at the plasma membrane to sense the environment, but you still reuse the same protein to do something else.
Yeah, and we don't know what that something else is. And we also don't know in a lot of cases what's already been shown. Where is that signaling coming from? And we have some ideas about assays that we might do, RNA sequencing and things like that, to try and identify transcriptional outputs from one location or another. But those are a little bit down the line. We haven't done any of those experiments.
And they're not easy either.
They're not, no. Especially when you want to look at signaling from different compartments of the same receptor, and even in some cases where you can do that, it's been done for example, the beta-2 adrenergic receptor, everybody's favorite positive control, for a while now. You can add in endocytosis inhibitors and look at the effects of signaling in with and without endocytosis. But for a receptor that's constitutively internalizing via non-canonical mechanisms, you can do it, but it's a little more difficult.
Yeah, yeah. And then if you want to throw in another monkey wrench, timing is everything. So it's very difficult. So not only it signals from two different compartments to do something, especially it's signaling from the endosome, but you don't, you also have to take into account as to the activation of the receptor if the pH at the extracellular site changes, but also at the internally. So what I'm trying to get to is timing is everything, and maybe depending on what's going on and when, the receptor has a differential function.
Yeah, and it could be very different from a lot of receptors. I mean, the kinetics of different receptor, how and when they see their agonists is varies a lot. But you could imagine that for certain hormones or say postsynaptic receptors, it's relatively short. It's a few seconds, a few minutes, a few hours, whereas for an environment sensing receptor like this, maybe it's weeks. And the difference between what the signaling outputs look like when you look at a receptor that's being activated over the course of weeks versus one that's being activated over the course of seconds or parts of seconds, I think there's a lot of room for more specific investigation there than at least I've done so far.
No, I agree with you, and I think in the context of proton sensing GPCRs and cancer and immunology, I think it's a more of a chronic change in the pH in the environment of the receptor. It could be a tumor microenvironment that'll cause, and I think cancers and the immune diseases actually, or immune deficiencies, take advantage of this and hijack this. But I find it fascinating that the endocytosis mechanisms are completely different from your classical, beta-arrestin mediated endocytosis.
Yeah, still working on identifying what they actually are.
I think it's really cool. Then comes my next question, which should be an easy one. What's your favorite GPCR?
Yeah, I mean, up until a year or so ago, I feel like I would have had to say beta 2, out of hometown pride. But I do think that I've gotten very excited by the work that our lab is doing on the proton sensing receptors in GPR65, and just generally the field of these, I don't want to say orphan receptors, because at least this one we kind of know the agonist in there. I feel like there are a couple of other ones, including one that I might be working on in the postdoc, fingers crossed, that we know a little bit about what might be activating them, but they're certainly understudied, and they're still known by their GPR whatever designation, and a lot of them seem to have either some amount of either constitutive activity or ubiquitous ligand presence, lipid sensing receptors and things like that, where it's not necessarily that they're constitutively active like on its face, but just that they always have access to these lipid ligands. So I think that these kind of classes of receptors, where it's maybe a little bit less about, they're activated by a hormone or something like that, but the trafficking is the main modulator of their function or the expression level. And so these slightly less conventional, at least by the canonical adrenergic, opioid, dopaminergic type of regulatory mechanisms. I think those are the ones I'm most interested in at the moment.
I love it. I love it. All right. I think we kind of discussed the current status of the research of your favorite receptor, and knowing that you just completed you just completed your PhD recently, and then you're moving on to a postdoc. I know you can't speak of it just yet, but I was thinking of towards the end of your PhD, when deciding what next, how did you go about it? What were some of the criteria that you had in mind to decide what's next for you?
I did think that I wanted to continue working on GPCRs, so looking for GPCR-based labs was important to me, mostly just because between having the training in it and having the tools to start asking those questions already, I wanted to some extent, be able to hit the ground running with questions that interest me. And then I also think that because of the huge number of GPCRs and the fact that they regulate pretty much every system, I didn't think that there was any dearth of questions that I can ask that will be interesting to me either now or in the long-term future. So, I think it just it made sense to try and stick in the GPCR field. But at the same time, I did want to move into a system that was a little bit more physiological, a little bit more therapeutically oriented than what I've done to this point. I think I have a pretty good foundation in basic cell biology, in HEC293 cells and some other cell model systems and everything like that. But I was really hoping to find a place where I could move into cell types and cellular systems and potentially, whole animal systems that have a little bit more depth to them, a little bit more complexity. And for me personally, geography was also a factor. I have a partner. She's living somewhere, and I was hoping to try and come and join her there. And so I'm actually very fortunate that I've been able to find a couple of different labs who I would be very excited about, with that meet my criteria. And then the other thing is I want to be somewhere where I feel respected and valued, and that I can get along well with the people in the lab, and I enjoy going into work on a daily basis because I like the people around me, both my lab mates, my mentor. I was really fortunate to have that environment throughout my whole PhD in general. I really liked, I was going to say the people I worked with, the people I still do work with. I'm going to go there in an hour or so, and you work with these people. But I've always felt that very lucky to have the lab mates that I have, both as direct colleagues and when I was starting as mentors in the lab. So, getting to know the people in the lab, getting to know the mentor, their mentoring style, and make sure that it works well with how I work, how I do science is important to me. And I think so far I've been very fortunate with that.
I love it. I love it. All right. Top 3 aha moments that you had as a scientist up until now.
Yeah, so I do think I'm still pretty early career, but
when I had to interrupt, you learn something when you make your agarose gel, or your gel with water and not buffer. So that can be an aha moment, because you only do it once, especially when you put on the most precious samples you've been working on all week, and then everything diffuses in the gel.
Yeah, I do think that to some extent, for me, science is a little bit less about the aha moments and versus the steady progress. Not everything's going to be moving forward, but for me, the moments that I'm excited are generally about, I'll have been working for a while on a project, and sometimes I'll feel like I haven't made a lot of progress, but then I will actually sit down with my data and think about what I have done and what I know now compared to what I knew a month, 6 months, a year, 5 years ago. And then looking back over the course of what I've done and realizing just how much I've done and just how much I have learned. So I think that for me, the steady progress is more salient than specific moments.
I like that. I think you're the first person who gives this answer, which I think it's a really unique answer. And you're right, in some sense, where being able to sit down and look at the progress, and being able to say, okay, when I started, this is what we knew in the field. Now, a year in, here are the things that I did contribute to. And to your point, when you said in the beginning that although it sounds like, there is this idea that academia is slow. It is slow, but when we keep on at it, we discover things and we establish, things around GPCRs or any field for that matter. And that requires time, that requires tools, and it requires people like you who are passionate to go into the lab and run those experiments and try and think about them in a larger context. Maybe today, taken apart or taken alone, this experiment is just an experiment for a PhD student, but you never know what the results will lead you to.
Yeah. And I do really like getting back big data sets. So, occasionally I've done proteomic, interactome studies with proximity labeling or co-immunoprecipitation. But yeah, I will say that it's fun to get back a big data set and see a huge tranche of data. I think that those are, specific, points where I can say, like, now I have all of this new information that nobody has seen before, and I get to be the one who sorts and organizes it and starts looking through it for what is interesting here. So I do think that, in the future, I'd be interested in continuing to do some of the types of experiments that involve RNA sequencing or proteomics, because I think that you start getting these big data sets, and as we get better tools and better analytical methods for sorting through them and comparing them, I do think that those are going to continue to be important sources. But then you got to actually go through and find something interesting. Like, once you found something interesting, you have to validate it. You have to go back to your standard workhorse assays. You have to go back to your signaling experiments and your protein-protein interaction assays and things like that. But I do those have kind of been aha moments when I get like a big new data set and I can start sifting through it for something that nobody has ever found before.
Speaking of tools and data sets, and for GPR65, if you had a magic wand, what kind of tools would you make appear in order to help answer some of the questions you have?
I would love to have one high quality, high affinity, water soluble activator and one inhibitor. Those would be nice. Those would be very, very useful things to have. It is a little bit, we've been able to do good work, I think, but it would be much, much more useful to be able to acutely activate and inactivate the receptor with something that I just add into the media. But
yeah,
we're getting there.
That's great. I forgot to ask about GPR65. I'm definitely no expert on it. Other than the constitutive internalization, does it activate your classical G protein, or does it preclude beta-arrestin, or what does it do from a signaling perspective?
It does seem to generally, work through canonical pathways in most cases. It activates cyclic AMP signaling. I will say, without going into too much detail, our lab does have a little bit of data lying around, again, from Dr. Morales-Rodriguez, that there are some really interesting things going on about exactly how it activates some of its signaling pathways, but it definitely couples to G proteins. There's been some work that it does so with some amount of promiscuity. It can activate a couple of different G proteins. We see a strong cyclic AMP response that is not there if you knock out G alpha S. So in general, its acute signaling appears to be fairly standard, at least at the first level.
Okay. Now, the reason I was asking that, because you mentioned, tool compounds. So that would mean that having a readout that's fairly reproducible, it would allow you to screen either virtually and then test in the lab, but be able to actually run assays and measure some activity, which is really, really fantastic. All right, last two questions. What advice do you have for your peers, colleagues, anyone interested in GPCRs or master's, PhD, or even a postdoc in the field?
I think something that's been very, very valuable to me has been just actually go to all the seminars that you can. Go and just hear about other people's research. I've been inspired to keep working when I am occasionally a little bit down and a little bit discouraged by seeing other people doing exciting science. So that's something that, in addition to being a little bit of a break from my own work once in a while, keeps me enthusiastic about doing experiments, is seeing somebody put together, a really exciting presentation of their own work and something that they're really enthusiastic about, and then, kind of stealing that enthusiasm and applying it to my own work, and then also learning about things that are outside exactly what I am working on, because there've been a lot of instances, both in the GPCR field and pretty much every other field of knowledge, where you get some significant advance, some real innovation by taking something that is, maybe really well known in some other field, but we, like, the people that we talk to, just either don't know about it or don't think about it that much, or just it doesn't come up that often. And so you can take those sorts of standard, everybody knows that in some other field, and apply it to a new field, and all of a sudden that's innovative. So yeah, read about and go listen to other science outside of the immediate GPCR community, because there's a lot of great work out there and a lot of enthusiastic people, and I think that it'll be helpful for our own field as well.
Love it. No, and you're right. Maybe there's one, it gives you a break from your own, let's say, quote unquote, biology questions or GPCR problems whenever you're working with the receptor, but at the same time, it might allow you to think about techniques and methodologies that you can import into the field and exploit. And I think it goes both ways as well.
Oh yeah, definitely. I assume that the people working on receptor tyrosine kinases are also excited when they see the GPCR talks. Although our department is a pretty GPCR heavy department here at Michigan, and I imagine at least some of the people outside of it are, maybe a little bit tired of GPCRs are the largest class of transmembrane signaling proteins in mammalian cells. They're characterized by their 7 transmembrane structure.
Yes, and then they are the target of to 30 to 40, 50% of FDA approved GPCR drugs, which is the I think any GPCR talk, these are the 2 first slides that you can see. All right, and last question, what should we title this episode? So you saw me typing, and I was actually taking notes as to what's your favorite GPCR, what's your aha moment, and what the advice is. I think the biggest question today is, what should we title this episode?
I will admit to forgetting to actually prepare for this specific question, so I'm trying to think as I play for time here.
Yeah, it's actually very difficult to answer the question ahead of time, because it also depends on where the conversation goes. So if the if, for example, today's conversation was, I want to say, more science focused, and we talked a lot about your projects, what's known, what's unknown, what are the things that you're interested in, and also about your career, next steps in your career. But there is sometimes episodes where we only talk science, or where we never talk science. So it really depends on, at the end of the podcast episode, is what was the focus of the conversation.
Yeah, I suppose it was very science focused, but I do think that, going back to something I mentioned at the beginning, was that, like, when I was starting my undergrad, I wasn't sure if I was going to do chemistry and biology or ancient Greek history and literature. And then, at the end I mentioned, go to the other seminars, see the other, see other people's talks. I've also still been really excited to be in a place where I can go to talks in the history department or something like that. So I don't know, something about, listen to the other fields, pay attention to pay attention to the universe of what's out there.
I like that. I had also an idea. I'll type this and I'll share that with you. What's out there? All right. So the title that came to mind as you were speaking now is something around the choice: ancient Greek chemistry or GPCRs, which I think is pretty much because you did have all of these choices and then you picked GPCRs in the end.
I did pick GPCRs, and I'm probably going to stick with that if I can. If people will pay me to do that.
So then the choices between what I just said or what you mentioned, pay attention to what's out there. It's a tough choice. I've been doing this with some of the guests lately, and it's a tough choice because there's both titles are good, but then at the end of the day, if you were to see this, I don't know, on social media or in your email, which one would you click on?
That's fair. And I think that you, as the experienced podcaster and someone who has developed an extremely useful and important hub for the GPCR community, I will leave that choice up to you and probably defer to your judgment on what plays best and what attracts engagement.
Yeah, because I think the discussion was really fantastic, and we need to make sure people really want to listen to it. And I feel like with all the information that you see around you, so not only in science, but there's social media, there's LinkedIn, there's Twitter, or X, whatever it's called today. It's hard to get through. So I'm hoping with the title, I think when people see ancient Greek chemistry and GPCRs, they're going to be like, "What? What is this podcast about?" All right, please go ahead.
Oh, I was going to say that sounds great. I'm sure that will be a lovely title.
With your permission, then I think we're going to stick to that. I feel like and it's been, again, it's a challenge. One of the reasons why we added the questions here at the end of the podcast is because normally the way the protocol goes is I finish recording the podcast and then I title it. And in the beginning, I used to give it titles based on the Zoom transcript, but we're not using Zoom anymore. And then it was based on my impression of the conversation. But I said, well, the team was asking, so Yamina, why don't you ask the guest? And we can have a conversation about the title, because you never know where the conversation can go. All right, Ian, thank you so much for your time. I really enjoyed our conversation. Good luck with everything.
Thank you very much. I also really appreciated the chance to come on and chat with you. It was lovely meeting you. I really appreciate everything that you're doing with the podcast and the webpage and everything.
Thank you so much. Don't go anywhere.
Okay.
I need a favor. If you enjoyed this episode, please click the like button. And if you like the podcast, consider subscribing to our YouTube channel. Many of you come back to watch our videos but aren't subscribers yet. One click goes a long way. All the information about Dr. GPCR is in the description. Find all our fantastic features and everything you can benefit from becoming a premium member. And while you're on it, check the Dr. GPCR University courses and share them with your network, colleagues, and friends. We also want to hear from you. Is there anything missing from our ecosystem? Can we make it better? Email Yamina at drgpcr.org or email our team at hello@drgpcr.org. Thank you so much for joining us in this podcast episode. If you're still there, we also want to take a minute to thank our team members: Yamina, Attila, Montserrat, Cam, John, Yatsu, Mark, Ivana, and Andreina. If you like our podcast, you can leave us a 5-star review on Apple Podcast. Until next time, and stay safe.